Hello, everyone. I'm Thomas Hermans. I'm the project manager for the JEDI COVID Grand Challenge. And so welcome to all the panelists that are going to be speaking today. Just wanted to give a brief update of how the challenge is going. So we have a really amazing number of teams that have signed up. So we have 118 teams at the moment that signed up. About half of it is from Europe, 23% from the Americas, 20% from Asia, 5% Africa. And of those teams, about 70% are university teams and 30% are companies and NGOs. So some very important information. So we have just sent out a new progress report. It's a type form. It will only take you 5 or 10 minutes to fill out. And basically we want to know what is your progress, the number of molecules screened, etc. And we have finally decided on the final deadline of the submission of stage 1, which will be the 17th of July. It's Friday at 6 p.m. Central European time. And we will shortly update the website of the challenge to reflect that. The reasons why we are giving such a big extension is that, it's definitely a very hard problem to crack the numbers of molecules we're asking you to screen. And we have had still a large number of teams joining in the past weeks, and we want to give them sufficient time to produce good results. So that being said, we have selected a few teams to give a progress update, which will be 5 minutes for each team. So please stick to your time. And these were a bit selected random. We had a number of applications, so we could not have everyone speaking, but maybe we'll have future opportunities to do that. So with that, I will give the word to Ashutosh Kumar, who's a researcher of a team that is called Kyuken, or I don't know if I pronounced it correctly, between the Riken Institute and the Kyushu Institute of Technology in Japan. So Ashutosh, I give the floor to you.
Yeah.
If you can.
Hi. Thanks, Thomas.
Hi.
Yeah. I hope everyone can hear me. Hello, my name is Ashutosh Kumar. I'm a member of Team Kyuken from Kyushu Institute of Technology and Riken in Japan. So I will be briefly presenting updates of our work on JEDI COVID-19 Grand Challenge. As the goal of this challenge is to screen more than 1 billion compounds. So we have accumulated around 1 billion compounds. And most of the compounds are from Zinc-15 database. Apart from Zinc-15 database, there are compounds from Merck library. There were some antiviral compounds from CAS. There were some drugs from KEC database, as well as drugs and herbal isolates from Sweet Lead database. So we have selected 3 protein targets based on their importance in viral replication as well as viral entry. Our first target is a protease, SARS coronavirus 2 protease 3CL-Pro, which has a role in viral replication. Our second target was the protein-protein interaction between ACE2 and spike protein, specifically receptor binding domain of the spike glycoprotein. And our third target was another protease, PL-Pro, which also has an important role in viral replication. So we have utilized a combination of ligand and structure-based virtual screening to screen a 1 billion compound library. Initially, we have developed ligand-based virtual screening models utilizing SARS coronavirus datasets. So we trained 2 classifiers and 1 regressor. And based on these regression and classification models, we were able to identify about 770,000 compounds, which were further screened using 3 separate virtual screening approaches. In the first approach, we calculated the shape similarity of these 770,000 compounds with the recently published alpha ketomide inhibitor. And apart from the shape similarity, molecular docking was also utilized using two separate molecular docking program. And the catalytic pocket of 3CL Pro were utilized. Finally, all these four scores were utilized to prepare a ranked list of about 10,000 compounds. For second target, the ligands targeting the ACE2 and receptor protein binding interface were not available. So we have developed a ligand-based model to recognize a generic protein-protein interaction inhibitors. For that, protein-protein interaction inhibitors were developed, and a classification model was developed using in-house classifier method. About 2.5 million compounds were selected at this step, and they were again further screened using three virtual screening approaches. For this target, we didn't know where we need to perform molecular docking. For that, we performed computational fragment mapping calculations so that small molecule binding hotspots on the surface of ACE2 protein can be identified. And indeed, we were able to identify a hotspot near the binding interface of ACE2 and spike protein. So this hotspot was utilized for molecular docking calculation using two separate molecular docking programs. And as well as the fragments which were identified using these fragment mapping calculations were also utilized to generate a query for shape similarity search. So a simplified query from these fragments were utilized to calculate shape similarity between these 2.5 million compounds. And finally, these three approaches, shape similarity and two molecular docking approaches, were utilized to create a ranked list of 10,000 compounds. For the final target, the virtual screening was more or less similar to 3PL Pro, where initially ligand-based virtual screening models were developed using in-house classifier. And again, SARS coronavirus data for PL Pro were utilized, and about 4 million molecules were selected, which were again filtered using shape similarity and two molecular docking approaches. Here, the shape similarity were based on a recently published peptidic inhibitor of PL Pro, SARS coronavirus 2 PL Pro, and molecular docking was performed at the catalytic pocket of PL Pro. And finally, the ranking, final ranking was prepared using the scores from these four virtual screening approaches, and a ranked list of 10,000 compounds were prepared. So we have almost finished the screening. So right now we are in process of writing the report as well as preparing the results for final submission.
Okay.
So, if you have any questions, please let us know in Q&A. Thank you for your attention.
Okay.
Yeah,
thanks a lot for your contribution. At this moment, I don't see any questions, so we'll move on with the next team. Thanks for your contribution. So this will be Team Nairo from Nairo Research in India. This will be Girinath Pillai. Sorry if I will unmute. Right.
Am I audible?
Yes, you're good.
Oh, thank you. So, good morning, everybody. Good morning, good afternoon. We are trying to explore with the team name Nairo India. We have few members. I have mentioned them in my last slide. So here we are trying to address something that we want to look more into selectivity, specificity, and sensitivity. Because as you can see, this is taken from Trends in Microbiology. There's a good similarity between SARS-CoV and SARS-CoV-2. So selectivity is also an issue. But at the current scenario, we need not to worry too much about it because it's just CoV-2, not about CoV. But still, when we are doing something, let us do it with a complete understanding. So we were trying to focus on understanding the different targets available. And this is also taken from genomic organization on different levels, which targets crystal structures are well defined, including RBDs, RDRP. So there's like a mechanism versus inhibition at different levels. So these are the different drug targets we are looking at. So our ultimate goal is that, as I understand, JEDI, one of the criteria is that we have to go with 3 different targets with billions of molecules. So we will be choosing the best 3 targets based on these 3 criteria which I mentioned about selectivity and specificity. So right now, once we send out the report, of course, there will be much more understanding and interpretations will be given there. There are drug candidates already being looked at on clinical trials and many more others. So we pick up a few of the protein targets based on the quality of the X-ray structure, as well as consensus on the co-crystal ligands, and trying to understand how far we can define the problem statement. So as I already told you, the mechanism versus inhibitor. At different mechanisms, we are to be very selective which inhibitors will be very selective to that mechanism. Or do we need to challenge and go with this multi-targeted approach? But it is too early to comment anything on that. It is more challenging, and it is a need of the hour. So based on that, we are trying to look at target discovery, height identification, lead optimization, ADMET. I understand we are more looking at virtual screening and identification, but why not? We can do some kind of optimization if it is possible. So enrichment factor is also considered based on virtual screening studies that were carried out, because there we do not want to have any false positives. And diversity versus scoring is also considered. It's not only about scoring will lead you to a lead molecule, it's about diversity also. So keeping that all these things in mind, we started a strategy with a deep learning based hit identification on 1.3 billion molecules, of course taken from Merck libraries, Smallport, Zinc, SCAS, antiviral databases, and many other data sets. So we made a selection criteria to bring in the best one, because computing resources are also, it's a time taking process. Then we did a virtual screening, considering binding energy, binding affinity, clashes, and enrichment factor. But here torsions are also being considered, so that we can ensure that the docked process that is generated could be a stable biological conformer, which is already present in some other X-ray structures. And we also explored with the hit optimization, like a hit triaging. Here we gave more importance to diversity, and then lead optimization, where we gave more importance to the properties and the binding affinity. Therefore, diversity and the scoring both are being considered for the selection criteria. And finally, we look at synthetic feasibility and accessibility if we come up with any novel analogs or derivatives. Otherwise, we just based on the diversity and scoring, we will select some of the molecules from the data set itself. So that's all. And for these different areas, there are few companies really supporting us, like Syslica, Optibrium, Biosolvite, and Pharmacera. So we are in the preparations of a report for these 3 different targets, and we will be keeping updated with the deadline on the submissions. And I really like to thank all my members and few other group members who are supporting this work. Thank you so much.
Thank you so much for this update. We'll do a couple of questions after all the 3 talks. So with that, I would like to move to the last flash presentation, which is Team SARS Wars, presented by Professor Travis Wheeler. Please go ahead.
I will be happy to share my screen when, there we go. When it lets me. Okay, I think that should get us to a place where you can see my Team SARS War slide.
Yes, perfect. Go ahead.
Okay, so thanks for giving us a chance to talk about our team and the pipeline we've been developing. The timing of this talk, I'm sure all of us who have now given this talk will feel that it's maybe a bit premature. We're all still working on pipeline, and like everyone else involved in the competition, we're incentivized to maybe not share all of the secrets of how it works just yet, but maybe those will give a sense of where we're going, what we're up to. Let me first describe the team that's working on this. It consists of Amit Roy, who's a computational structural biologist at Rocky Mountain Labs, NIH Research Institute in Montana, and Vish Venkatraman, who is in Trondheim, Norway, at the Norwegian University of Science and Technology. He specializes in chemical informatics, and then myself, Travis Wheeler, and 3 grad students working in my group, Daniel Olson, Connor Copeland, Jeremiah Geyser, where we come from sort of the computational genomics and machine learning realm. And we've come together to develop an computationally efficient and maybe novel drug discovery pipeline that we hope will identify compounds useful in the battle against SARS-CoV-2 and beyond that, other viruses we'll encounter in the future. First, we all have to pick drug targets. Our motivations for picking them were that we wanted to identify proteins that had no mammalian homologs to avoid risk of cross-reactivity, and that for which there's good structural modeling. And we've identified nucleocapsid N protein, the RNA polymerase NSP12, and the main proteinase 3CL-Pro. These will probably not be surprising to those who have been working on this project. The approach we've taken it involves a pipeline that maybe differs from what we've heard so far. Rather than beginning with a machine learning approach, we began with a prediction of the shapes of pockets for these target proteins, using FTMap and POCASA and some manual curation, identifying a number of pockets for each of the proteins that we'll use as binding targets, and with those pockets engaged in rapid prediction of ligands that appear to have good estimated fit. That's a custom method that we'll talk about one of these days. And we'll call these predicted ligands seeds. And we've used those seeds to serve as a basis for trying to identify a collection of candidate ligands that are synthesizable and to explore a much broader range of possible ligands that bind into our pockets, culling through the set of databases that I've listed on this slide here. It's a pretty broad set. I think everybody will recognize that this is basically the set of all the molecules we're likely to be able to enumerate. It ends up being something almost near 4 billion unique ligands that we're culling through to identify ligands that are similar to the ones that we've predicted as being decent candidates for binding into our pockets. The pipeline follows up by identifying fingerprints for the seeds that we've predicted, plus these 4 billion candidate ligands using mechanisms in the RDKit framework, and then an in-house method that we've developed to rapidly search for neighbors of our seeds from within these 4 billion sets. And this is based on the Tanimoto coefficient using some SIMD instructions for speed. And the result of this search has been to identify about 100,000 what we'd call close neighbors to our predicted seeds, and in total about 1 million that are somewhat farther. And we're interested in this full set of a little over 1.1 million, or a little under 1.1 million neighbors. To give a sense, among those close neighbors, we've explored around 40,000 targets for NSP12, 35 for N, and 20,000 targets for the typo there, 3CL-Pro. Within that set of close neighbors, I think it's useful to just have a sense of the enrichment of which of these large databases seem to be relatively decent sources for what we think are maybe viable drug targets, and the top three are data sets that are known to provide drugs that have been used in the past, and they're Sweet Lead and Drug Bank are relatively enriched. 1 out of 500 of the entries in Drug Bank made it into our final close neighbors set, 1 out of 2,000 of the Sweet Lead, and the rest of them range on the order of 1 out of 10 to 1 out of, call it, 100,000 of the candidate molecules is made it through our pipeline to later analysis. The pipeline follows for this set of 1.1 million candidate neighbors with a quick filter to remove ligands that show risk of toxicity, the ADMET filter, which doesn't really filter all that many of them out. It's under 10% of the candidates. And then we engage in in a predictive docking approach, which identifies multiple candidate docking poses using standard software Autodoc Vina. And it can be tempting to just say, great, Autodoc Vina identifies a pose that has a good estimate of binding, and one of the mechanisms for identifying that, or for evaluating that pose quality, is a measure called DFIRE, which it turns out those who have been looking at this probably know this, is not a particularly good predictor of binding affinity or of an active and decoy or true positive and false positive binding status.
Time's almost up. So
this is my last slide, so we're great. And what we've done is we've taken these pocket estimates, and we've developed a deep learning estimate of binding affinity, trained on a couple of data sets, DUDE and lit PCBA, and taken efforts to avoid the bias and decoys that are a problem for this field, and are currently engaged in the final stages of classification and ranking, using Nina and a custom model internally that we're developing to identify an enriched set of active compounds. Thanks for your time.
Thanks, Travis. Just a question to the three speakers that were just talking. How do you get only a ranking out of your results, or how close to a binding affinity to a dissociation constant can you get with your methods? Travis, maybe first.
Ask me in 3 days.
Okay.
We're in the midst of moving from the prediction, from a classification prediction to a binding affinity via regression model, but I wouldn't describe it as reliable yet.
Okay. Ashutosh, can you comment on your math?
Yeah, our score is more or less like a ranking score, so it's not as accurate as a binding affinity predictor, but more like a rank list. So it's a normalized score, so we cannot compare with it the actual binding affinity.
Okay. And Girinath,
yeah,
can you comment?
So ours is like a multi-parameter, so that it combines score, binding affinity, clashes, that is intra and interligand clashes, as well as torsions.
Okay. Another question that's coming in is how do you take synthetic feasibility into account, if you do? Do you have thoughts about that?
Yes, we are looking at the smart filter where the bond breakage could happen and bond forming could happen. Now, it's a prediction, right? So I cannot say that is going to scale up to experimental also, but yeah, predictive capability, yeah, it is fine. Feasibility to some extent, at least on the prediction stand, yes.
Okay. Ashutosh, you can comment?
Actually, like some of our compounds are like commercially available, but like many of the compounds are make on demand compounds. So the vendors, they guarantees that they can synthesize 80% of those make on demand compounds. So
I saw on okay.
Yes. So most of them are make on demand compounds.
Okay.
So
Okay,
I think
thank you.
it depends
Yeah.
on vendor.
Right. I saw on Travis' slide, I mean, you had 1 in 200 from the sweet lead. So, I mean, those are existing compounds, but some of the other lists you pointed out, like, I mean, clearly not the bottom ones where you're in the billions, but which of those libraries, or what's the percentage of compounds that you estimate needs to be synthesized for Travis, for Professor Wheeler's project?
Oh, the large majority of them would need to be synthesized, but we've done an assessment of synthesizability, and almost all of the compounds that have made it through our initial filters show as being likely synthesizable. 95% or something. I don't remember the exact numbers, but a large fraction of them.
Okay.
Which was encouraging and surprising, actually.
So there's a question from Craig Baumann. What are the inputs and outputs for deep learning methods being used? I don't know who wants to take that one.
Shall I?
Sure.
Yeah. The inputs we have taken is collection of these targets, as well as the collection of the library compounds, which were being pre-screened with certain filter criteria. These were the input, and the output was a ranking criteria and the threshold criteria, which is being set based on the benchmarking.
Okay. Greg, you can actually.
Yeah, I'm just trying to see, like, is it actually doing docking of sorts? So it's taking structure and ligand structure and somehow guessing at affinity, or
it's just smiles. It is taking no structures here.
okay.
So in our pipeline, we've moved we've put the deep learning classification step later in the process, so post docking prediction. Mm-hmm. So we're performing docking with AutoDocVina and gathering a collection of docked poses and number of features for each of those poses, and dumping those in as a set of input features, along with some extra features that are universal, not specific to any particular pose or dock, to predict both classification and, we hope, a regressed.
So like with that, you've trained it on a bunch of complexes to like find interesting combinations of features, and now you're looking for things that have. Okay.
That's a good high level description.
Yeah.
Okay. Maybe last question that came in from the attendees is, what is the lowest docking energy you have achieved so far? Ashutosh, you want to?
Yeah, it depends upon the target as well as the docking program. So suppose like we utilize Fred and Gold. So suppose like the average score for our target is -10. So I think like with screening billion compounds, so you increase the probability of getting a lower score. So we have achieved, I think, like -17, -18 for Fred. And similarly for Gold, if the normal average score is around -8, -9, so we have achieved around -15, -16. But majority of the compounds are still around the normal average range.
Okay. Thank you so much. So I think with that, I would like to wrap up this first point. So thanks to the three teams for taking the time to present. And then we will move on to the invited speakers. And the first one is Craig Baumann, who's actually leading the Folding at Home initiative, which is a very interesting crowdsourced computing initiative that is really working very hard on SARS-CoV-2 issues. Craig, the floor is yours.
Thanks. All right, is my screen showing up for folks?
Yes.
Yes.
All right, great. So obviously all the experimental structures that have been coming out so rapidly are immensely valuable, and very generally my lab is interested in, well, what else can we learn from the rest of the ensemble of structures that a protein adopts, and what new therapeutic opportunities might that open up? And so one of our particular curiosities has been about cryptic pockets that are absent in these experimental snapshots, but that we might find if we could watch how all the moving parts of these complex machines go about their business. So we've been at this for a while, and I won't go into too much detail, but we have a whole suite of methods for very directedly hunting for these things. And so this is just one of the enzymes we've been working on, where I've labeled the active site where this thing performs chemistry. So that's the site that most people have been targeting from a drug design perspective. But if we watch the snippet of a movie we pulled out based on our analysis, you can see this other binding pocket opening up that was not at all obvious from that initial structure. So we've got a number of experimental assays that we've adapted or developed to test for the existence of pockets like this. And we've moved into trying to use the structural information about these pockets that we get from our simulations then, especially once we've experimentally corroborated the presence of a pocket, to start designing small molecules to bind these sites and potentially act as allosteric modulators. And so to take into account all this conformational heterogeneity, we have a very simple method we call Boltzmann docking, where we have a network representation of our protein's energy landscape on the left, where you can think about it as clusters or states or energy minima, where the protein tends to, structures the protein tends to adopt. What we do is dock compounds against representative structures for each of these states. Often we have thousands to tens of thousands of them to get fine-grained structural resolution. And now we can calculate a Boltzmann-weighted average score across this ensemble and rank our compounds by that, which is great because it's going to do things like naturally favor more probable structures, for example. So we've got a couple of interesting hits. So now we're looking down on this protein. Unfortunately, my mouse doesn't show up on this slide. So the active site is kind of in the middle of the protein. And at the bottom left, you can see that cryptic pocket where these helices have opened up, creating this unexpected binding site and pose from our docking with a compound there. And so we've shown that this compound, as well as two others, actually allosterically modulate the enzyme's activity. And then if we mutate away key contacts that these compounds form, then experimentally the compounds no longer have any effect on activity, corroborating that they really bind where we're predicting here in this novel pocket. And interestingly, we found both inhibitors and activators that bind this site. So that's an interesting aside that opens up fun possibilities for the future. So we've done a lot with these enzymes, and then in the recent 2 years, we've been looking at some other targets to see what we can find. And I won't go into great detail, but this is a small protein from Ebola virus that plays in a really important role in suppressing a host's immune response. And in the closed structure, you can see when this closes up, that's what's been seen experimentally. And there aren't really good sites to target with small molecules to inhibit the multiple interactions that this domain forms with viral and host factors to control the immune response. And what we found is this very interesting cryptic pocket that now again, we've got experiments to corroborate that this actually exists in in the solution in ensemble, and has many features of a druggable site. We were just got our first preprint up on this, and we're gearing up to apply our drug discovery methods when we got a little bit distracted by stay-at-home orders and the pandemic. So especially thinking about viral proteins, it didn't take long for us to think, we should turn all of these tools loose on SARS-CoV-2. So we and other labs involved in the Folding at Home project, this massive distributed computing project, where it brings together now over 1 million citizen scientists who volunteer to run computer simulations on their personal computers to accelerate our ability to get at long time scales in many systems, for example. Before we started this, we had about 30,000 devices participating in this distributed computing project. Now we have quite a lot, so well over 1 million, and so it's a huge computational resource. And so the first thing that we wanted to do was look at the spike complex, so these red protrusions from these very common pictures of the virus. And this was really interesting to me because these viral complexes use a fascinating conformational masking strategy where the conserved interface of these proteins that actually binds to the ACE2 receptor on a human cell and initiate infection. And the structure that we see, that's the dominant structure of these things on the surface of this virus, that's actually buried in the core of the complex of 3 proteins that forms each of these red protrusions. And so it's really clear that this thing has to open up in order to engage a host receptor. And I think very little is actually known about how this opening occurs and what the open state looks like. And our thought that it would be really awesome if we could get at that with these tools and maybe identify potential ways of targeting this crucial component of the virus that's so highly accessible on the surface of the virion based on that structural information. And so here is a view of that. Most of that complex. So the upper left is tethered to the surface of the virus, and the lower right is where the 3 receptor binding domains, or RBDs, are, but where the interface that engages this ACE2 receptor again is buried. And so by bringing all of this compute power together on this very large system with minimum 400,000 atoms, because this is like 3,000 residues or 12, together, we're able to see these really dramatic motions where down here you'll see one of the RBDs opening up and becoming accessible and available to interact with ACE2. And so we're excited to get this out there and start delving into what we can learn about this process and which of the stages of motions like this might be interesting therapeutic targets beyond what one could do with just this closed structure, for example. All right, so this is a big system. It's taken a lot of compute power, but we have, like I said, a pretty insane amount of compute power. Our estimate is that we're the first to reach the exascale, so at least 5 times more horsepower than the largest publicly available traditional supercomputer. So in addition to looking at the spike, we've been going after everything that we can, everything we can build either there's an experimental structure or we can build a reasonable homology model. And when I tell people about this, they often have lots of reasonable questions about, like some of these things form oligomers or complexes. Are you simulating monomers or complexes, or, which variant are you looking at? My kind of joking response to this is just yes. Like, we're trying to do everything we can in parallel, and ultimately to get the data out there for other people to mine as well. So we've got something like over 100 different simulations of variants of SARS-CoV-2 or SARS proteins, and some of the host factors they engage with, and some of the complexes they form, and so it's a really fascinating data set. So just as another example, we've been starting to look at the main protease. So here up on the kind of upper left is the active site. This thing also dimerizes, but my understanding is that the affinity between the monomers is weak enough that it might be interesting to also try to disrupt the dimer by stabilizing the monomer. So we've got simulations of both the monomer and dimer going, and this is coming from the monomer simulations, but we've actually found a couple of interesting cryptic pockets, the most dramatic of which is this swiveling of these two domains that creates a reasonably sized groove in between them that one might be able to go after as a means to disrupt dimerization, given that our understanding is that the dimer is the active form. So my lab has been focused a lot on these conformational changes, but there's a number of labs involved, and in particular, a shout out to Vince Voles and John Cadera, who have been doing a lot with binding free energy calculations, especially through the COVID Moonshot, which I think we'll hear some more about. Normally my impression is that it's common for people to run, order of a dozen, or a couple dozen calculations. They've done, I think, as many as 70,000 different compounds now. So it's a really fascinating scale to go at with these, very intense methods and see what value we can extract. So that gives a flavor of what's there. We're working hard with a couple partners to try to get the data out in the public. I'm hoping that some of the first data sets will come online this week, but it's been a slow process getting things in place to get data sets on this scale out there. But have a really great team working on it that I'm very, very grateful to, and a lot of support from different organizations, making sure we can stay on top of this explosive growth and have the best chance of extracting some value from it.
Great. Thank you, Greg.
Yeah.
Are there any questions from the panel or from the attendees? I was just so the spike protein actually kind of opens up and closes. So what's the implication for the connection with the ACE2 binding domain?
Yeah, so the issue is that our immune systems are good at finding like conserved surfaces, right, and learning to recognize those. And so what this thing is doing is, in its dominant state, it's burying those conserved surfaces, and now the surface that our immune system can generally access is like covered with glycans or is more mutagenic than other surfaces. So it's harder for our immune systems to find patterns and build up immunity. But of course, it still has to have these conserved interfaces to successfully engage with human receptors. And so this conformational masking strategy is burying that in the dominant state of the system, but having it exposed some fraction of the time so it can productively or unproductively, from our perspective, engage with a receptor. So
Okay.
I think there's interesting opportunities both in, you could imagine trying to capture the closed form to prevent it from opening up. You could imagine, targeting novel, structural pieces that, you see in this opening motion that you wouldn't guess at from this motion, right? So for example, when this thing is highly open, people have found antibodies that bind deep down in here in regions of the protein you wouldn't guess were accessible if you thought that mostly it adopted this conformation and just harmonically wiggled around that structure.
Right. Okay. Well, we'll have some more time in the Q&A after. So with that, thank you again, Greg, for your wonderful
My pleasure.
insights. Then I would like to give the word to Professor Stan Whatwich from UTMB, who has been in contact with us and has been sharing very interesting insights on a big initiative as well that is going on in Texas.
Thank you very much. Delighted to be here and to meet with everyone. Let me see if I can find my screen. Just 1 second. Let's try that. So we decided to take a slightly different approach even before knowing about the challenge, with the idea being, should we even get involved in a COVID-19 antiviral project, or is this just a distraction? And so for the youngins on this call, you may not remember back in the early 2000s, the US had a big anthrax scare. And then there was a big effort by the government for biodefense. And every scientist, here's a pot of money, let's go for it. And at our University of Texas Medical Branch, we got a level 4 national lab built, designed for infectious disease, and everyone was really excited. And then BU, Boston University, was trying for this. And then in typical fashion, the funding started to go down. And it's actually very challenging to keep these national labs going. And then, we had the first coronavirus outbreak, and everyone got all excited and, let's jump on this. And there were some funding efforts, and then it petered out. And then, we had MERS came out, and we should get on this. And so when COVID-19 came out, the question we really asked our friends, neighbors, colleagues is this something that we really want to get involved in, or is this a distraction from the science we typically do? So as we tell our students, don't go chasing money, follow the science you really care about. And so with that thinking, we said, all right, we will only get involved in this project if we, one, think we can do something useful, and two, if we can do it rapidly. So given how fast the virus was spreading, at the middle of April, we said, we'll start a computational effort to look for novel therapeutics to add to the pot of therapeutics that are being proposed, but only if we can get this thing done by 6 weeks. Can we do a screening project, come up with compounds, get them tested against virus, and check cytotoxicity in 6 weeks? That was the challenge we set. Otherwise, we said it's not really worth our time. All right. Well, here we are in June, and we're about 3 weeks behind schedule, and I apologize for that, I guess. So this group got together. We had some online meetings, and the goal was to see if I can go to the next slide. To say, can we? Let's see if I can go to the next slide. Let's just go. Let's go bold. Let's go for every. So we're going to do a structure-based drug discovery approach, where we want to get these compounds that the computer predicts into testing ASAP. Now, all of you on the call who have been doing structure-based or any sort of computational chemistry work, I hate to say it, we don't do it well. No disrespect to any of us, but if my graduate students came to me and said, Wadowich, I want to run this gel, and about 5% of the time, on a good day, it really works. And what I get the results that we're expecting. And I'd say get out of my lab. But on a good day, the computers are predicting, and we can argue about this over beers. But on a good day, the computers are predicting maybe 5% of the hits show some activity. We've been doing this for a large number of years, and that's kind of what we see, regardless of what programs we use for structure-based drug discovery. VINA, Glide, yada yada yada, even consensus. So we knew this was going to be a high-risk project. So we said, let's go for all the structural structures that we have for COVID out there. We decided not to throw in the helicase, where we just had a homology model. And of the 6 non-structural proteins for SARS coronavirus 2, there were several structures other than for the M protease, where there are 80-odd structures. So we just picked a handful of the M protease structures. But for all the other ones, we said, all right, what's out in the protein data bank? Take all the chains for these structures. We ended up with 17 structures, 17 unique chains, proteins, and we said, what are we going to screen against? And this was before we saw the JEDI challenge. We just said, we've been doing this long enough. We want to get answers quickly. I do not have a stable of medicinal chemists that can synthesize 10,000 compounds quickly, especially if I tell them, look, you know, synthesize 10,000 compounds and maybe 5% of your efforts are going to be productive. That never makes me friends. So we said, we're going to screen against libraries that we can immediately order compounds. And the biggest libraries out there that we felt were relatively well curated were the NMN library. So we spoke with NMN. We took their 3 libraries. They had a premium collection of about 46,000 compounds, an advanced collection of about 450,000 compounds, and there's what they call their high throughput screening library of about 2.1, 2.2 million. So we took all of those, combined them, generated the various stereoisomers. We're left with about close to 6 million compounds that NMN claimed we could order immediately. So that was the goal. So again, the big picture is screen, high risk. We know there's going to be a lot of nonsense that comes out. Order the compounds, get them into the screening centers. See if anything comes out that would advance the effort in a real short period of time. So we had been running, you know, VINA docking programs at the Texas Advanced Computational Center on their supercomputers. So their team of computational chemists put up the NMN libraries in a few days, and we quickly screened all 17 different proteins against the libraries. Then we did a second round of screening, taking the top several thousand hits from VINA, rescreened them using GLIDE. So the Schrodinger package GLIDE. We decided not to do a whole bunch of other, if we had more time or if we wanted to take more time, I'd be interested in trying to do a very large consensus project and develop a consensus score, but we just used 2 parallel virtual screening programs. We had seen in the past that they give complementary results. We made then a consensus ranked list from those for each protein and for each chain. We selected, we ran them through the usual light pains filter and ADMET filter, got rid of several compounds, and we're left, and then we chose about 100 ligands per protein target. In this case, this would be a particular enzyme. So about 600 compounds, because we talked to NMN and they were gracious enough to say, sure, you know, give us a couple plates, you know, give us 600 compounds you'd like, we'll give them to you free. Don't tell them I said that, but they were very generous. There were some hiccups in getting the compounds ordered from NMN, number of holidays, a number of closings that made transporting compound libraries on plates across the world a little more challenging. But the plates were, by the end of June, excuse me, by the end of May, we had the 600 compounds from NMN delivered to Texas A&M in Houston to do cytotoxicity screening, and to Boston University to the level 4 lab up there where they had robotic facilities. So Rob Davey, colleague, runs the high containment lab up at Boston. Cliff Stevens runs screening facility at A&M. So within a week of receiving compounds, the first week of June, Cliff's team had finished the cytotoxicity screening in, they were using, in this case, Vero cells. So we know what compounds. They did a dose response curve, 6 different doses. Of the compounds we gave them, about 20 appeared to have some high levels of cytotoxicity. So Rob's group have run into a little more problems. They finished their first round of antiviral screening last week. They weren't satisfied with the reproducibility of the controls. And so before giving us the results, they want to redo the screening, which they're doing this week. So again, we're about 3 weeks behind where we want to be. The end of this week, we should have the results of the antiviral screening. Again, we realize very high risk, because in addition to just simply trying to find a small molecule that inhibits a protein enzyme activity, we're doing this in cell culture. So you have the added joy of the compounds need to be soluble. We know many of these libraries have compounds that aren't soluble in DMSO, let alone buffer that's compatible with cell culture. The compounds have to get into the cell. They have to cross the plasma membrane. So we're going to lose compounds there. So it's a high risk, but the idea was, can we do something very rapidly? It's a little different strategy than the great effort that the JEDI group is doing, which is again looking for compounds that might be unique inhibitors for proteins, testing them against the proteins, the ones that work, then move forward. So our goal is to say, all right, anything we find from the antiviral screen combined with the cytotoxicity screen, we'll then move them down to the Galveston National Lab, do follow-up, hit validation in a number of different cell lines, and then move them into the animal model, there's a mouse animal model that we have in San Antonio that the compounds will move into. We hope that that could happen by the end of July now. So that's, in a nutshell, what our strategy and focus would be. So we hope to get everything posted early next week up on the JEDI Challenge site. Again, high risk. It may not work, but we figured would give it a shot because we had the resources to do it. So that's our approach. So thanks very much.
Thanks a lot, Stan. I'm just is that a common approach, or do people traditionally always go through the first identify the protein target, the binding, then moving slowly down the line? So you're making a big jump now?
Yeah. So we're making a jump from doing it, shall we say, more systematically, where you'd say, all right, let's look at a ligand, do a biochemical experiment, ideally, some sort of, whatever your biochemical assay of preference might be, even something as messy as an IC50 for enzyme activity, find something that seems to work, it's going to work poorly, and then make analogs, do your KCO2 activities, et cetera, et cetera. And again, we do this all the time in many experiments, and it can take months and months and months, and then you need to bring on the medicinal chemists. And so again, for this project, it was designed to be very rapid to see could something of this approach with a very shotgun method. If we see anything, then we'll expand it and screen additional compounds. So again, I don't think any of us has a magic way of doing it, which is really unfortunate that we don't. But is this going to lead to what we all hope for? Is a United Nations Institute for Rapid Drug Discovery and Development that can respond to any sort of threat? Yeah, one can only hope, right? But I doubt it.
Okay.
Maybe in Europe, where you guys, no disrespect to America, but maybe in Europe where you might have a longer term vision that this needs to be done. Again, one can only hope, or maybe the Gates would do this, but not seeing this unified long-term effort to really get this done well. I know we have, Dr. Brooks on the call. He's been making great strides for many, many years, and we're still not at the place where we can, accurately predict binding energies. It's a really challenging project and problem. Mm-hmm. So let's get it solved one of these days.
Yes, exactly. Okay, I have some more questions, but I'm going to save them for the Q&A after that talk of Alpha Lee. So thanks for now. And then I would like to move to Alpha Lee, who's a PI at University of Cambridge and also co-founder of Postera. And they're leading the COVID Moonshot, which is a very interesting project, and hoping to hear more of that.
Thanks so much for having me on the call. I think I've just managed to unmute myself.
Yep, you're good.
Cool, great, thanks. So in my talk today, I want to discuss our progress in COVID Moonshot. This is joint work with Frank von Delft, John Codera, Neil London, as well as many, many others around the world who kindly contributed their time and effort in the Moonshot. a brief, I'll first give a brief overview of what Moonshot is, and then I will discuss our progress, and finally I'll talk about several highlights and where do we see this initiative going forward. So COVID Moonshot is, in a nutshell, a crowdsourcing exercise for an antiviral. It's all started with a tweet by Martin Walsh in Diamond Light Source, where he tweeted the very interesting work of a fragment screen against the main protease of COVID-19 done by the group of Martin Walsh and Frank von Delft. And then at that time, I was in the US, part of new startup Postera, which aims to bring sort of machine learning with medicinal chemistry together to accelerate drug design. And we thought, here are a lot of very promising fragments, and it would be great to bring these fragments together, very naively perhaps, and design novel and potent inhibitors from these fragments. And in particular, our idea is twofold. The first is, back then and still now, there are a lot of medicinal chemists working from home in industry and in academia. Therefore, we thought, well, there are these huge amounts of fragment hits against MPRO. You would be able to get a strong inhibitor, presumably, by joining these fragments together or merging them. Why not open this up as a challenge or as a community exercise of just joining these fragments together? So it becomes a crowdsourcing initiative where we told the participants and everyone around the world, hey, here are the fragments. We got 71 fragments bound to MPRO, some covalent, some non-covalent, and please design inhibitors based on these fragments by joining them, merging them, or expanding them, right? And just by doing that, we thought we might get 100 submissions or 200 submissions, but actually we got 7,000 submissions from over 300 scientists from academia and from industry. So that's great to have a lot of submissions. But then the next question is, well, how are we actually going to realize these submissions? Because these are sort of a directed set of compounds that are designed according to fragments. And that's where Postera's technology and the technology we've been thinking about comes in, which is in the design make test cycle. We think that design is something that a lot of us has been very excited about, both computationally and otherwise, but synthesis is always an effort that is incredibly time consuming. In fact, in this, if you want a rapid response and being able to make compounds quick is incredibly hard. And that's where we deployed large scale machine learning technology, which predicts which compounds could be made easily from commercially available building blocks. And these building blocks are increasingly functionalized and complex. Therefore, you can realize quite complex chemical space if you think about which building blocks to buy and how to join these building blocks together. And so we combine this technology together with crowdsourcing to power the whole Moonshot. And in short, top line summary, we found 25 compounds with IC50 less than 10 micromolar, and we recently found a 500 nanomolar inhibitor that has been confirmed with 2 different assays against MPRO. It started with me, Frank, John, and Ner, but has now since the whole initiative has since gone worldwide. We have Pharma, UCB at Boehringer Ingelheim participating, Enamine contributing their time, Psi contributing their time pro bono or at very low cost, and it is a genuine global effort. So what is unique about COVID Moonshot? I've given you a high-level summary of it. First, it is a completely open exercise. All of our compound structures are deposited online. There's no IP claimed or involved. All our participants agree to share everything. International teams are working pro bono at cost. I've outlined the few main institutions, but there are many, many more. We have 300 submitters around the world. A lot of them are from academia and industry. And importantly, I think we target, rather than targeting necessarily the design stage of the design make test cycle, instead, at least from Postera's perspective, we target the synthesis stage of the design make test cycle. And we squarely focus on how to, rather than trying to think about necessarily computationally generating data, we want to experimentally generate data. And so if we think about experiments, then there are 2 parts. First is synthesis, the other is assaying. And so we use machine learning to accelerate synthesis by deciding which compounds could be made quick and how to make these compounds. And that means, for example, we can triage over 2,000 compounds and found synthetic routes to them in less than a weekend, which would take an ordinary chemist maybe a few weeks to go through thousands of compounds by hand. And also partner this with advances in high throughput assaying, in particular, high throughput crystallography techniques pioneered in XChem and Diamond, which allows us to gather very rich structural information really quick. And we can therefore go through hundreds of compounds, all with not only IC50 measurements done in Neil London's lab, but also with high quality crystal structures done in Frank van Delft and his collaborators' labs. A little bit more about the synthesis technology that we contributed as Postera and as my group in Cambridge. The idea of this synthesis technology is basically learning the rules of chemistry using natural language processing. We basically deploy machine learning models to learn the outcomes of chemical reaction given reactant and reagent by just mining all the reactions that's been published in the literature, and in particular in patent literature. And we have published this recently, and it is the first model to outperform even trained human chemists in predicting challenging chemical transformations, including regio, stereo, and chemoselectivities. And we repurpose this model to do retrosynthesis, and that allows us to rapidly disconnect molecules that participants have submitted into commercially purchasable building blocks. And we can therefore assess whether the molecule is makeable, and if so, how long will it take to make the molecule, and therefore make a realistic assessment of how many molecules can we shove into the pipeline to be made as soon as we can, yet still exploring diverse chemical space. A brief sort of technical summary of the results that we have. We have now assayed over 500 compounds and identified 4 promising series which we are pursuing. We have 2 non-covalent series of quinolone and the amino heteroaryl, and 2 covalent series based on the Ugi chemistry and aryl piperazine choroacetamide. 2 of the series have broke the nanomolar potency threshold. One is covalent, therefore you may argue is cheating, but the other is non-covalent, and that is what we are actively pursuing right now. And the reason why we are able to, again, explore diverse chemical space, we also make sure that compounds we push in the pipeline be rapidly made, rapidly delivered, and rapidly assayed. And to give you an example, this compound has only been assayed IC50-wise a few days ago, and we already got a high-quality crystal structure of our 500 nanomolar binder bound to the binding site of MPRO. And I think it is these sort of rapid experimental iteration enabled by synthesis technologies, but also obviously aided by computation efforts. For example, what Folding at Home has kindly contributed to us, which allows us to power design tests as fast as we can and aggressively shave off cycle time. And we have already assay cascade set up for in vivo testing. If you want to find out more about Moonshot, please visit our website. One of the key premises of Moonshot is that all data and all insights are shared openly. We don't hold back anything. For example, here's a discussion of the different binding sites in MPRO, and you can find sort of weekly annotations of the data coming from Moonshot. This is our annotation, a commentary of our data from last week, where we discussed the latest crystal structures and the MedChem team's thinking about where to go next in terms of exploring the binding site. And towards P5 and towards new areas binding pocket is an area where we want to go next. In terms of what are the next steps, we have already aligned live virus assays, RAP-PK studies, and thinking about planning the whole pipeline towards in vivo studies, all with pro bono efforts coming in, helping us. I think this is a team of pro bono, open pro bono scientists working together, and we have been very humbled by the support that we have received. And ultimately, we aim to build a drug hunting ecosystem for pandemic preparedness, and several of us has written a brief some commentary on that published recently in Nature Chemistry about hopefully a open science initiative can perhaps be a way whereby data can be parked, efforts can be shared, and that could build towards a community where we find new drugs to prepare us for future pandemics. And that's the end of my talk, and looking forward to answering any questions. Thanks so much for having me.
Thanks a lot, Alpha. Okay, I'm just trying to go through some questions. I mean, it's of course a very important problem, on how to increase this pandemic preparedness and how to streamline these things. But one question maybe to both Stan and to you is, okay, now you have some results that you will put out in the open. How can we convince other initiatives, other people, other companies to take these things downstream?
So I think there are precedents of open drug discovery. For example, DNDi for neglected tropical diseases or neglected diseases in general. They have actually done clinical trial on drugs that have been fully disclosed. I think pharma understands that there are diseases which requires a different model than the conventional business model. There's also another model, M4 Pharma, so the open source pharma model, whereby the IP is accrued at the very end in the clinical dossier rather than in the preclinical chemistry. And we think that with antivirals, in particular for infectious diseases in general, there is no profit incentives anyways to, I would argue, to actually develop anti-infectives, antibiotics included. And therefore, I think we would think a promising way perhaps is to make data available so that investigators can take the data and conduct trials and gradually accumulate enough evidence to convince stakeholders to conduct trials, rather than the conventional model of data and insights being protected via IP. Not that IP is wrong, but I think in this particular instances where the economics are firmly stacked against drug development for infectious diseases, perhaps another model is worthwhile exploring.
I would follow on that and say, with the large number of efforts going on, if that de-risks some of the early stage work, I think that's very valuable. Getting the compounds into animal models within kind of our own resources by tapping into our collaborators, showing efficacy in the best animal models out there is kind of the critical next step. And at that point, I think there's a chance to convince governments and philanthropic agencies such as the Gates comes to mind, but there are others, to spend the few $1 million that it would take to do the safety trials that are necessary, certainly for the FDA to accelerate into phase 1 clinical trials. And at that point, then the government would step in. But I think what we're all trying to do now is de-risk that early phase and get us into animal studies. Again, the efforts we're all doing are relatively low cost, but they're very, very risky. And so hopefully 1 or 2 of them or 3 of them work. And the next efforts, less risky, and a little more expensive, but given what the US government is spending on vaccine efforts, which are great, almost $2.2 billion for discovery efforts, not manufacturing, putting a few $1 million into safety, preclinical safety trials for anything that, let's say, Alpha Lee's group comes out with, or any other groups come out with, is a small investment. So I think that's the hope we have to all have.
Mm-hmm. Okay. Well, actually, I had a more specific question to Alpha, actually. So there are other algorithms, of course, out there that do retrosynthetic analysis. Actually, I did my postdoc in the lab of Bartosz Rybowski at Northwestern. That was one of the first developers that was now turned into the Cynthia software. But so can you highlight again what's unique about your approach and how does it compare to other approaches?
Yes, that's a great question. So I think the Cynthia approach, and in fact a lot of the synthetic route prediction approach, uses manual curated reaction rules, and I think Bartosz should curate a lot of these reaction rules almost by hand and use that as a premise to disconnect molecules. What we have found is that we can use machine learning to basically learn the nuances of chemical reactivity, but just directly training it on reaction data. We have curated over 10 million reactions published in patents and other sources. And we can basically just treat reactions as a language where the reactant and reagents are treated as language and the product treated as another effective chemical language, and it can parameterize more abstractly a machine translation model that goes from reactant, reagent to product. And that's so-called template-free approach where we let the model learn chemistry from scratch. And although that is usually a bad idea when we don't have enough data, if you have a lot of data, then the model is able to distill the nuances of chemical reactivity a lot better than handcrafted rules. And that's what we empirically found out when we sort of benchmarked this against a data set of expert human organic chemists predicting outcomes of chemical reactions, where we found that the model is actually able to make correct predictions about the products given reaction and reagent a lot better than even skilled chemists making the same predictions. Because the model has access to millions of data and it is able to learn all of it rather than we having to read the data and then distill heuristics from it. And I think that's the key technological advance that enables the model to do so well.
Okay. Maybe I'll give the word to Andrei Luskruk-Pietri, who's the executive director of JEDI, who has also a follow-up point.
Well,
Andrei.
Thomas, I suggest, hi everybody. I suggest we continue. We still have a bit of time for Q&A, so keep the word and we'll do the closing in a bit of time.
Okay. I'm just trying to wrap my head around still about this taking things downstream. So your, let's say, the vision, if I summarize Alpha and Stan's vision together, is that, you know, if we find good enough hits, that the governments will step in and actually save our butts. No, I think it would be great, of course, but well, I mean, I don't know of any.
I would say not save our derrieres, if I can use my my multilingual skills, but I would say with animal data, that is a big step forward, and we can collect animal data using our collaborator network and almost for very low cost and get either buy-in, et cetera. That data would be very convincing to, let's say, the NIAID, who is having programs out there. That would be very convincing to the US DARPA, who has programs out there looking for new approaches. And again, when you talk about going from animal efficacy data to then safety and manufacturing, we've done this with small molecules. It might cost, and again, we can debate the actual amounts, but $1 million to $2 million, and you can get that done in about 6 months if you really accelerate. Now that's not quick, and but it is relatively inexpensive in the scheme of all the money being poured in. So it's not that the government's going to save our behinds, but you would have very convincing data to tell the government, here's something that is past animal trials. It is looking very, and with that you could compare it to other things that are being proposed. So that's where I would say is that when you get that animal data, you've done one really big de-risking step. The next big de-risking step before clinical trials is safety. What's your safety margin? The good thing is you're talking about an acute infection where there's nothing else to treat, and so you may be able to tolerate a slightly lower safety profile. Maybe not. I mean, that would be an FDA decision. So again, I think it's all about trying to get that really important animal data as quick as we can. And people have talked about different approaches, from having batteries of cages of animals that you just screen your compounds right then and there. Okay, I'm not in favor of that, but this is, I think, one approach. And I think what Alpha is doing with his effort is, again, trying to see how quickly can they get towards serious animal studies, and that's obviously what we're trying to do too. And I think everyone else is. And it's, again, how quick we can do it. Alpha, what do you think?
Yeah, I agree completely. I think the key de-risking stage is the in vivo efficacy, and I think that provides a lot of evidence and confidence in de-risking this early part of drug discovery.
So are people working on kind of, it's not really crowdsourced, of course, but to collaborate heavily on a global scale on these animal models? Because still, I mean, if you look at, depends on which animals, of course, but if you look at non-human primates, the costs are incredible, right? Like a small trial is $1 million, or
No,
so
less
what's
than
the
that.
best?
Yeah, the animal trials now with the non-human primates that are being proposed are, again, depends on what one's definition of reasonable is, but tens, maybe $100,000 because, again, they're very rapid trials now. And that's, if you have to look for a silver lining, and there are many, but one of them is that we're dealing with an acute infection that, you can see the results very, very quickly. So you don't have a long-term maintenance cost, et cetera. So, and we do have other models without jumping to a non-human primate that work well. Not, again, it's not completely duplicating the very wide range of symptoms that we're seeing in humans, but you're definitely seeing the infections take off even in a mouse model that can be used to collect your efficacy. And the FDA has said, you don't need 2 animal models for efficacy when you start talking about some of these infectious diseases. So I don't think animal models, the testing phase is the limiting factor. I think it's doing the testing and having some really positive data to show for it. That's my feeling.
Yeah, I think, for example, our collaborators already have ferret models of infections up and running that's been repurposed for ferret's model for influenza, for example. And for ferret, for example, ferret or mouse or even dog, tox, that's a relatively standard thing you can outsource a CRO for toxicity, mini tox or full tox. So I think, yeah, the animal study ecosystem is relatively well established.
Okay. And I was surprised to see for Alpha, for the COVID Moonshot, that you have already assayed 500 or 600 compounds.
Yeah.
And so I'm just curious about is this coming directly from the crowdfunding, or how have you managed to do so many compounds so quickly?
You mean funding, or you mean
Well, both, yeah.
Right. So in terms of the funding, we got crowdfunding, but also the leading labs committed or repurpose existing resources. We also got a lot of philanthropic funding coming in through Oxford, through Boltzmann, for example, to support the effort. But I think in terms of the logistics and the fact that we know which compounds can we make quick, yet still complex enough to be interesting, allows us a substantial advantage, because I think folks like Enamine has collected a huge library of really complex building blocks. And if you can join these building blocks judiciously together, you can already build pretty good lead-like compounds in almost 2 or 3 steps. And that's like 2 or 3 weeks delivery time. There's no need to photosynthesis, make something that's crazy. And we found that the compounds which we really judiciously selected based on synthesizability actually use a lot of information, because it's a quick turnaround, and we can turn 3 or 4 cycles around in 2 months. And that gives us a lot of SAR. For example, the amino ARAL, hetero-ARAL series came from the aminopyridine series, which was basically a 2 or 3 step synthesis directly from inner building blocks. And once we saw the hit there, we said, okay, like it's already like a 10 micromolar hit. Then the further optimization, again, just using library type chemistry leads us into 500 nanomolar territory, all right, 2 or 3 steps. So
okay, thanks. Maybe I want to go back to Craig if he's still around. Greg, you showed in a simulation. Let me just check.
I'm still here.
Okay. You showed in one of your simulations, you saw actually the protein dynamics with the ligand inside of it, right? That looks kind of computationally very heavy. So what does, what does this exactly, is maybe it's a naive question, but what does this exactly add to the understanding of how this compound binds?
Yeah, I mean, so the idea is to, in a more rigorous way, account for the degrees of freedom, both within the protein and the ligand, right? So really what you want is the binding free energy, and any docking method that assumes the protein is rigid and the ligand is mostly in one pose is overlooking the whole conformational entropy aspect to this, and the potential role of solvent and whatnot. So this is using that particular. Most of the work so far has been with absolute free energy calculations, and I think John is getting relative free energy calculations up and running as we speak for comparing different compounds. So it'll be a really interesting one, right? Because in principle, it's taking into account more of the relevant physics, but at a large computational cost, and no one's been able to do an experiment on this scale. So it'll be a very interesting data set.
Mm-hmm. And is this a method that is it becoming commercially available? Because a lot of the docking softwares are, let's say, either publicly available or commercially available.
That's right. Yeah, no, it's all available. So all the absolute free energy stuff, like those methods are all in GROMACS. So that's publicly available. And, people, I know Schrodinger is doing a lot with related, development of free energy methods. And then the relative free energy calculations that John is working on are all in OpenMM. So both GROMACS and OpenMM are open source and available to anyone to go at. So.
Okay. Right. And if you look at the road from here to a therapy, Greg, like how do you see this developing?
Yeah, so I mean, we're pretty tightly integrated with Alpha and COVID Moonshot. So a lot of this, John and Vince Voles are both involved in this collaboration. So it's the exact same pipeline there. And we've got data for all these other targets too. So there we haven't done as much, if anything, with screening small molecules yet against all the other targets. So the idea is more to get the data out there, and then both we and others can think about how to put that structural insight to use.
Okay, maybe let's open up for wider discussion of all the other panelists. Anyone wants to jump in here? Okay, no, just one thought maybe for for everyone. But so Alpha, you've been crowdfunding your efforts. What about crowd synthesizing molecules? Like, can we recruit the synthetic labs of the world to reunite and help us out?
Oh, that's a great idea. So we didn't do that yet because most labs were closed during the pandemic. Now, obviously, the world is reopening. What we found is that logistics is a very interesting problem. Like I've just DHLing stuff from Pune, India to Kiev, and then Kiev, Ukraine to Oxford, and Boltzmann is a challenge. So when you have n different labs synthesizing things, you need to ensure purity are all of the same standard, etc. So that's why we've not yet done that. But I think if there are hubs which would be willing to commit a lot of resources in that I think will definitely help a lot. And in fact, I know the NDI has this open synthesis network, which we are talking to, and they are doing that model whereby they ask sort of universities to volunteer some synthesis resources.
Stan, do you have a feeling for, if this would be a good idea, or what people would be willing to commit from the academic side or from industry side?
I think from the academic side, most people are willing to commit a fair number. I think, like Alpha said, one of the things is logistics. Quality control is a major concern when you start talking about synthesis. Just the effort it takes to coordinate a large number of synthetic labs. It certainly is doable. If you say, all right, here's a list of compounds we want, if you have expertise on such and such, let's go for it. These are the parameters, and we want, and if you have then a central location to do your NMR, to do your purity checks, et cetera, so that you know what you get, it could be very valuable. I think on the scale that Alpha's team is looking at, when you're talking several hundred or thousands of molecules, it sounds like in-house they have that capability to do that synthesis relatively rapidly, and I give them lots of credit for having that capacity. And so I think, when you're talking in that scale, if you can keep it under one roof, especially when you get into the disruptions that we're faced with right now, is just transport, et cetera. I think it's a lot more efficient to do it the way, keep it all in-house, like Alpha's team is doing for some of these final steps. Alpha, what do you think your in-house capabilities would be for compound synthesis in terms of like a month? You generate probably several thousand compounds with your team, right?
Yeah, so just to clarify, so we don't actually do the synthesis in-house. We engage CROs to do it. So it's Enamine, WuXi, and Sci Life Sciences doing the synthesis for us. And the capacity there, I think for 3-step synthesis, you can make a 2-3-step synthesis, you can make a few hundred per month with a few FTEs. But obviously when you get the lead optimization, they really want to make the right compound, or you hope it's the right compound, then that becomes slightly more challenging. But I think for those complicated synthesis, you do need sort of very sort of dedicated, we are going to make that compound not because of an organic chemistry interest, but because of a medicinal chemistry need. And that's where a lot of these CROs have this professional drive in just like churning out libraries of compounds. And that's what we found particularly useful.
Yeah, so that's a great model, because again, as opposed to, duplicating efforts that CROs have already established, utilize their pipelines. I know WuXi STA claims they have 2,000 medicinal chemists on board today for a project that they can engage, and they know their capabilities. So yeah, leveraging what exists is a good approach.
Okay, I think in view of time, I will give the word to Andre. You're still muted, Andre. Right, there you go.
Well, thank you first to all the speakers, first to the 3 performers. Thank you for sharing. As Travis said, I mean, I'm sure you didn't share all your secrets, so we are very impatient to see the final results. And thanks also to Ashutosh and I don't know if our third is still there. And thanks to Stan, Alpha, and Greg. I mean, we are obviously doing something altogether very important. You saw how disappointing the current repurposing efforts of current drugs are. So discovering new compounds is obviously critical. I remember 2 months ago, we had a lot of discussion with some large foundations, which were very focused on this repurposing effort. I'm sure that has probably a little bit diminished. So we are really there. I mean, there's some worrying news about some second wave coming. So it's clearly very important. The second thing I take up, especially Stan, Professor Vartovich, your suggestion, and you give a lot of credit to Europe. and our long-term vision. Well, as you know, we're building the European version of DARPA, but on the civilian side. Yes, probably the acceptance, especially for scientists from all over the world, is higher here currently than on the other side of the Atlantic, but that will probably be only short term, hopefully. But I'll take that up from you, because I think maybe that there's a follow-up discussion that we should have with Charles Brooks, Alpha, Greg, and you, and others, of course, to see how, and that's my third point. I mean, the common thread of everything we heard is about disrupting the preclinical drug discovery process. And this is probably ripe for disruption. So I think this is what we probably could, with this crisis, there are some tectonic shifts. Let's use it to move and get our collective effort in that. We'll try to, I mean, I encourage also everybody who is following us, keep following us on our Twitter account. That's the best way to know what we are doing. We're going to have a tracker of how many molecules have been screened so far. So we heard several billion so far. So it's at JEDI. And for the final word, I leave you, Thomas, for the next steps, especially. So thank you to all of it. It was really exciting.
Thanks, Andre. So as a final reminder, so please have a look at the the survey that we sent out to all the teams, so we can track your progress. I
mean, can't insist enough on that.
Right. So really fill the survey.
How things are moving. This is what allowed us to also postpone a little bit the deadline. We want to make sure we have really breakthrough results. So it's more than tracking, is really to understand where, if we have as many teams as possible getting crazy results. Yeah, sorry, Thomas.
That's it. No, that's all I wanted to say. So the deadline, 17 July, 6 p.m. European time, and soon we'll also update you on all the steps that we will take after you submit your results, because there's a lot of challenges already. Some were highlighted by Alpha, but there's a lot of steps to be done to get to a real therapeutic treatment. So with that, thank you so much for joining us today. Thanks to all the speakers, all the panelists, all the attendees, and I wish you a great day.
Thank you. Thank you all.
Thanks, everyone.
Thank you.
Bye-bye.